I'm Jeanne Tie here at ASCo 2016, presenting the results of my study, the potential of circulating tumor DNA to shape the design of clinical trials testing adjuvant therapy in patients with early stage cancers. The use of adjuvant chemotherapy in stage 2 colon cancer remains a clinical dilemma, and better markers of recurrence are required. Here we demonstrated the ability of circulating tumor DNA to detect minimal residual disease and to assess the impact of adjuvant therapy. In routine practice, patients with stage 2 colon cancer are offered adjuvant chemotherapy if they have high-risk features, but this approach involves treating a large number of patients for modest chemotherapy benefit, and surveillance included regular CEA and CT scans. The conventional approach to testing the benefit of adjuvant treatment requires long-term follow-up of a large number of patients. The goals in the management of stage 2 colon cancer are to improve patient selection for adjuvant treatment and to detect recurrences earlier. Adjuvant clinical trials should enrich for high-risk patients with occult disease, incorporating biomarker providing early readout of treatment benefit. Our traditional approach to estimating recurrence risk involves the examination of resected tumor tissue for high-risk pathological features such as T4 tumors, presence of lymphovascular invasion, or adequacy of lymph node sampling. Clinicians then use this information to make their treatment recommendation. An alternative approach would be to examine peripheral blood taken after the primary tumor has been removed for the presence of circulating tumor DNA. The detection of circulating tumor DNA after surgery is a direct indicator of residual micrometastatic disease, much like how we would use staging CT scan to detect macroscopic cancers. Circulating tumor DNA are small cell-free DNA fragments released from tumors through the process of apoptosis or necrosis and are distinct from circulating tumor cells. Typically, mutations identified in the tumor tissue can also be found in the circulation. ctDNA can be detected in a majority of metastatic solid tumors, and recently small series have suggested a role for ctDNA to be a marker of minimal residual disease after potentially curative surgery in various early stage cancers. This large prospective multi-center study enrolled 250 patients. Tumor tissue from 231 eligible stage 2 colon cancer patients were analyzed for somatic mutations. Peripheral blood taken post-operatively and 3-monthly during follow-up were examined using a personalized assay for the same mutation detected in the primary tumor tissue. Adjuvant chemotherapy was given to 23% of patients at clinician's discretion, blinded to the ctDNA results. Surveillance included CT scans, 6-monthly and 3-monthly CEA. During follow-up, 15% of patients had experienced recurrence. The objective of the study is to demonstrate that the presence of ctDNA is a marker of minimal residual disease and to explore changes in ctDNA status during adjuvant chemotherapy were given and during follow-up. The primary endpoint was recurrence-free survival. At a median follow-up of just over 2 years, the majority, 11 of 14 patients who were ctDNA positive had recurred, compared to only less than 10% of those who were ctDNA negative, with a hazard ratio of 18. We also found that these difference between ctDNA positive and negative patients remain highly significant in the clinical low risk and clinical high risk groups. One of the potential advantages of a marker of minimal residual disease is the ability to track changes in tumor burden over time at a microscopic level below the detection threshold of CT imaging. And potential scenarios include those positive ctDNA which remains positive after completion of chemotherapy and continues to rise during surveillance, and those who had a continuous response to chemotherapy and during surveillance. We showed here some examples of ctDNA levels in patients treated with chemotherapy on our study. Those with a durable response in ctDNA remain recurrence-free, whilst those with persistently elevated ctDNA even after chemotherapy experience recurrence. So in conclusion, we found that detection of ctDNA provide direct evidence of residual disease in patients with stage 2 colon cancer, and this could further risk stratify patients to adjuvant chemotherapy as well as detect recurrence earlier than imaging. In clinical trials, ctDNA analysis may improve trial efficiency through enriching for high-risk patients and providing early readout treatment benefit. Thank you.